All right, everybody. It is Chapter 10, Part 2. Okay, there's a lot of information in Chapter 10. So last week, or last lecture, the types of cells, the specific vocabulary to skeletal muscle fibers was looked at, and we looked at excitation of the muscle and how to contract it with the three key parts. The neuron tells the muscle to be excited by releasing acetylcholine, and acetylcholine binds to its receptor on the motor end plate. And in fact, that's kind of going here. And then once acetylcholine binds to its receptor, sodium channels are open. That allows sodium to flow in. That's excitable. That flows down the membrane, down the sarcolemma of the muscle fiber, through the T-tubules, and releases calcium from the sarcoplasmic reticulum. Calcium then is able to bind to troponin. And by binding to troponin, tropomyosin is pulled off of the actin binding site, and myosin and actin then interact. Well, after they interact with ATP being present and available, then you will see that that the ATP will be expended and we will get a muscle contraction. All right, so that was a lot of what we went through in that first, the neuromuscular junction, the how to excite the membrane, what exactly is excitation, and then the sliding filament theory. Now we're going to go into how when that muscle contracts, so how when an individual muscle contracts, we call that a twitch. How can we make that twitch more powerful? How can we make it generate more force? All right, so it's going to be looking at specifics on, okay, we get calcium released, we get the muscle fiber to contract. Well, how can I make it stronger? How can I make it better? All right, and that's where we're going into length-tension relationships and trying to manipulate and make a better interaction of how the overlap of actin and myosin occurs. So when the muscle then is generating a contraction and changing its shape and producing a force, that force is more powerful. All right? So again, when fibers are going to shorten, because the myofibril in each of these muscle fibers, there's lots of myofibrils, and myofibrils are made up of sarcomeres. All those sarcomeres pull together and produce a force, and then we call that force a tension, right? And because one part of the muscle, as it shows you down here, because one part of the muscle is fixed and the other part is movable, the insertion site, all of that tension, all of that force production is pulling in one direction. And we can measure that amount of force, that change in pull, all right? And if we can manipulate situation to make this change in pull stronger, that is sometimes to our advantage to create a more powerful muscle contraction. Right? So that's what this next little lecture is going to be about, is how can I get, when I generate a muscle contraction, one individual contraction, so I get acetylcholine released, I get calcium out of the sarcoplasmic reticulum, and I get actin and myosin to interact. How can I make the sarcomere shortening? How can I make the pull that they generate on their insertion point, how do I make it stronger? Okay? And again, contraction is an all or nothing event. So acetylcholine being released causes that neurostimulation, causes free calcium in the sarcoplasm, causes actin and myosin to be able to interact, and as long as I have ATP available, I'm going to get that muscle cell giving me 100%. Every actin and myosin that can interact interacts, all the ATP that is available is expended, and the sarcomere slides and shortens, and the force production is produced. So I'm getting a maximal effort, but how is it that I can make that muscle more strong when it contracts? Okay, and some of it again, muscles contract with neural stimulation, and muscles are going to go into relaxation as the neural stimulation goes away, so there's no more acetylcholine in the synapse. There's no more acetylcholine binding to its receptor. Calcium then is going to start to slowly go from being available in the sarcoplasm, the cytoplasm. It's going to start getting pumped back into the sarcoplasmic reticulum, and then as calcium numbers are less, there's less calcium and troponin interacting, and then there's less actin and myosin interacting, and then as actin and myosin are less interacting, there's less ATP being expended and sliding filament theories occurring. And slowly that decrease in calcium, that decrease in actin and myosin, and some of the recoil forces of just the muscle wanting to go back to its resting tension is going to lead to the muscle to then go from a slided, smaller sarcomere back to a kind of elastic recoil back to original position of its sarcomere, and then the muscle fiber is going to go back to its resting length, its resting tension level. Okay? And so that is muscle contraction, that is muscle relaxation. All right? And getting a muscle to contract, you have to have a nervous system stimulus. If you cut the neuron, you can't get a muscle to contract. Getting the muscle to contract, you have to have calcium available. If there's no calcium, there's no contraction. Getting a muscle to contract, even if you have the nervous system stimulating, even if you have calcium available, if you don't have ATP there, no contraction. To get to relaxation, you have to get rid of the calcium. If you don't get rid of the calcium, you continue to stay actin and myosin interacting. You have to not have ATP available or stop making as much ATP, so that way actin and myosin again are not having a plethora of energy. If you keep letting the neuron be stimulating and releasing acetylcholine, again, you can't get the muscle to relax. Right? And relaxing, there's some intrinsic kind of cytoskeletal proteins that want to return the muscle to its original length, and so the muscle wants to, upon relaxation, go back to its resting place. All right? And that resting place is such that there's a certain amount of actin and myosin overlap. And so when I start talking about, well, how can I make that muscle generate more force? One of the things I want to try to get the muscle to do is start maybe at a different sarcomere percent of overlapping between thin and thick filaments position. And in order to do that, to get the relaxation to maybe open up a little bit to a longer sarcomere, I need to maybe put a tension or a pull or a weight on my muscles. And so that's the first part of, again, trying to get actin and myosin to maybe interact for a longer period of time and more interactions, is getting the resting tension, instead of starting with this much overlap, to start with less overlap. Okay? So this is just a summary side. Again, the three critical pieces of getting a muscle to contract is the neuron talking to the muscle releasing calcium, calcium and ATP, actin, myosin interaction. Getting it to relax, acetylcholine is taken away by the enzyme acetylcholine esterase. So the acetylcholine is chewed up. It's no longer binding to its receptor. As it's no longer binding to receptor, the sarcoplasmic reticulum is going to start to pump and pull the calcium that's in the sarcoplasm back into the sequestration of this reticulum. Without calcium, tropomyosin is going to push back onto the active site, so myosin and actin can't interact. And then just through the natural recoil of the proteins, they're going to go back to a position where there's a little bit of overlap, but not as much overlap as was occurring with the contractile sliding filament theory. Okay? So, getting a muscle to contract, you do those. Stimulate it, make calcium available, get ATP available. Getting it to relax, take away the acetylcholine, the calcium, and the ATP. Now, when I want a muscle to contract, and I want it to generate 5 pounds of pressure, that's great. But what if I need it to generate 5.3 pounds of pressure? I need one muscle fiber, one cell, to give me a little bit more force. Okay? How can I make a muscle cell that has a certain resting tension, a certain amount of overlap, a certain amount of actin and myosin, a certain availability of calcium, a certain availability of the ATP? How can I make it generate a little more force? I go back to that sarcomere and I think about how can I make the sarcomere and the areas of overlap a little bit more optimal for getting more force production. And for most of your muscles, their resting length of what the sarcomeres are sitting at in actin and myosin overlap is not actually the most ideal place. So most of your sarcomeres are sitting to where if you ask them to produce a certain amount of force, they will. But if we can get the sarcomeres to stretch a little bit and get the amount of actin and myosin overlap to be a little less, I now am setting up the sarcomere to, when asked to contract, have more availability for actin and myosin to interact more times, more forcefully, and generate, instead of 0.8 units of force, to make 1 unit of force. Okay? So getting the muscle to generate a little bit more force in a single muscle, single contraction, part of that can be achieved by taking the muscle and its resting tension and its resting shape and try to stretch it a little bit so the sarcomere has a little less area of actin and myosin overlap. And so it has a little more space then when it's asked to contract to shorten and cause more actin and myosin to interact in that process. And more actin and myosin and more ATP expended means more force production. Okay? And so what this graph is trying to show you is that the optimal place to get the most force production out of a sarcomere is not usually the resting position. It's usually a little bit longer than what the muscle intrinsically wants to rest at. If we let the muscle start with no overlap between actin and myosin, so it's stretched too much, we get no force production. And if we let the muscle start with its sarcomere fully overlapped and the thin filaments are all the way to the middle, then there's no force production. So sarcomeres need to have some level of ability to slide. So we don't want them to be so stretched out that actin and myosin don't intersect, and we don't want them to be so crisscrossed and overlapping that there's no room to go any further. And in many cases, the muscles tend to sit with a resting tension that is not in the ideal position. And so getting them to be a little longer, getting them to be stretched a little bit, is going to enhance, when you ask the muscle to produce a tension, to produce a force in a given one twitch scenario, is going to help you produce a more powerful twitch. Okay? So now take, in an organ, I don't have one muscle fiber. I actually have hundreds of muscle fibers. So if I get the organ level to not have just one muscle fiber generate a little bit more force production because I stretch one muscle fiber, but if I get every muscle fiber to then be a little stretched, I'm actually going to get the muscle to then maybe produce a little bit better in one contraction, because every muscle fiber contracting is doing a little bit better at force production. I'm going to get a more powerful force. Okay? So that's one of the ways I can make a muscle contract in a stronger, more forceful way when I look at one muscle cell, one contraction, and the amount of force it puts out. All right? Now, the second way to get a muscle and a skeletal muscle fiber to give me more force of contraction in a given force output is to mess with the frequency of stimulation. Okay? So, to understand this, we got to take our muscle cell out of our body and we got to put it in a myograph, this experimental design where I have a, I'm going to try to draw this for you. All right? So, I have one muscle cell, okay? And this one muscle cell is attached to a device. So here's the muscle cell, and it's attached. So this end is attached to one hook, and this end is attached to one hook. And I have a little piece of nervous tissue, a little electrical signal that I can send down and cause, kind of like acetylcholine would, an excitation on the muscle fiber. All right? And when I tell the muscle fiber I just push a button, and then when I push the button, the electrical energy travels down my wire onto my muscle fiber, and that muscle fiber then allows the calcium inside the muscle sarcoplasmic reticulum to be released, and then the calcium gets on troponin, and then actin and myosin interact, and I have ATP available, so ATP is expended. I can then measure the pull, so the change of one of these hooks. So it might be that when the muscle shortens, it changes this height, okay? So when I put all of that on a graft, I am going to get a Myograph, all right? And again, a Myograph looks like this. So from the point in time I push the button, I am going to see there's a little bit of a delay, and the delay is going to be represented with the time it takes for the energy to go down from the button, down the wire to the muscle fiber, and release the muscle fibers calcium out of the sarcoplasmic reticulum, and then the time it takes for calcium to bind to troponin. All right, so all of that is represented with the latent period. And one of the things about the latent period in this Myograph experiment is the latent period is always the same. So if I put a muscle from my arm, if I put a muscle from my face, if I put a muscle from my hip bones or my skeleton, my butt, the latent period, the time it takes for me to push the button, and the time to then see the muscle start to contract. And so I know then when the muscle starts to physically change its shape, calciums bind to troponin, actins and myosin are interacting, sliding filament theory is occurring, and I'm expending ATP. I know that that's the time it took for the energy to get to the muscle and calcium to be released. That latent period is always a fixed time, and in many of the experiments it's like 0.2 milliseconds. Okay? Now, in this Myograph, once I start seeing, again, calcium bound to troponin, tropomyosin's off of actin, actin and myosin are interacting, I can then see a force production. And again, going back here, if the force production starts with no room to overlap, I'm not going to get a lot of force. If the force production has a little bit of room to overlap, I'm going to get a little bit of force production. If the force production is in an optimal position, I'm going to get the maximal amount. If it starts to be not quite, stretched out a little bit too much, I won't get as much force production. And if there's no overlap when I start, I won't get any force production. All right? And for most of our muscles, they start and they sit here. And so getting a little bit of stretch actually puts them in the more optimal position. All right? And that's one way to get this force produced to go up, is first off, start the muscle in a different resting position. So start the sarcomere in a different place. The other way to get that muscle to go into a higher maximal setting is going to be to mess with getting another stimulation or contraction to occur either exactly at the point of the twitch ending or before it ends. Okay? Now, sometimes you're going to hear about muscles are going to twitch fast or slow. All right? So when we take muscle from the quadriceps, the hamstrings, the biceps, the soleus muscle in your calf, the eye muscles in the eye, the muscles around your lips, and we do a individual, every muscle fiber is looked at, we see that not every muscle fiber twitches the same amount of time. And that's what this graph down here is trying to show you, is that this amount of force that a muscle with a certain number of myofibrils, a certain number of sarcomeres is going to produce is pretty much going to be an all or nothing response when acetylcholine or when the electricity button is pushed in the myograph experiment. But because muscles are going to have different amounts of time that they spend twitching, some muscles are going to be fast twitch. So if we look at the eye muscle, the latent period is the same for all three muscles, from the eye and the calf and the gastrocnemius of the calf and the soleus of the calf. Always 0.2. So that's the amount of time the electricity takes to get from pushing the button to the muscle fibers membrane, gets calcium released, calcium binds to troponin. Okay? But when we look at the time that the eye muscle spends with actin and myosin interacting, so the amount of time that calcium is available, actin and myosin interacting, and ATP expended, it's less than 20 milliseconds. And so that muscle goes through a complete release of calcium, actin myosin sliding, twitch produced, force production produced, and then back to relaxation and ready to do it again within 20 milliseconds. The muscles from the gastrocnemius, so the nice big head of the calf, that muscle, 0.2 milliseconds for latent period, but it produced a twitch, a force production, but it took 40 milliseconds. So it's a little longer. And the soleus muscle, the one that's underneath the big fat muscle of your calf, that is really important for walking, it's a slow twitch muscle. So it takes almost 100 milliseconds to get that muscle fiber, to get all those sarcomeres to completely contract and then to completely go back to resting relaxation state. So get all the calcium back in the sarcoplasmic reticulum, get all the troponin, tropomyosin back on top of the actin, and then the actin and myosin to go back to their resting sarcomere length. Okay? And so these myograph experiments, one, they let us learn about how long it takes most of the muscle cells of the body with their latent period to get calcium released when they're excited. It lets us see that most of the muscle fibers as an individual cell produce a certain amount of tension, and it's pretty much a certain amount given that they're doing their 100% effort for whatever the sarcomere starting point and number of overlap and actin, myosin, and sarcomeres and myofibrils produce. And then it tells us how long it takes for that entire contraction and relaxation cycle to take place. How long it takes, if we go back to this slide, how long it takes for all of this and all of this to take place. So the time it takes to get it all to contract and then all back to resting level. Okay? So that's what these myographs show us. All right? And so one of the things we learn with these myographs is that different muscles have different times that it takes them to cycle through excitation and relaxation. Different muscles, again, because of the different number of sarcomeres and the resting starting point, are going to generate a certain amount of force production. And we can make that force production maybe go up a little bit the next time we ask it to twitch by manipulating first the starting point of the sarcomere. But the second way we can start to mess with the contractile force produced in a single twitch is to start messing with frequency of stimulation. Okay? Now, to understand how frequency of stimulation is going to affect, we need to, underneath the twitch, underneath the curve, define periods of when the muscle is building up its force production. So that's the period of time that the actin and myosin are contracting, and we're getting increase in actin and myosin interactions. And then when you start to look at the second half of the curve, that's when we start to say maybe some of the calcium's being taken away from troponin and put back in the sarcoplasmic reticulum, and the number of actin and myosin interactions, the number of overlapping and ability to slide further is just not as great, and so we're seeing less force production, and so we call those the relaxation phase. Okay? And so when I want to get this contraction to maybe be a little bit more height, one of the things I can do with messing with stimulation frequency is start to try to get my second stimulation on this muscle at a timeline where it either happens the minute the muscle is finished contracting, while the muscle is in its state of relaxation, or I start to let the frequency happen while it's maybe even in its contraction phase. Okay? And so getting a twitch then to go higher in force production, 1, mess with the sarcomere resting length. 2, mess with the frequency. Okay? Now, when I mess with the frequency, there's a few things I can do. The first way I mess with it is I do what's known as Trep. So Trep is defined as I make a contraction happen at the exact time point that the muscle that just contracted is relaxed and done with its cycle. So if we go back to that previous slide here, if I want to see Trep in the eye muscle, I want my frequency rate to be every, let's say, 13 milliseconds. So I am going to ask the eye muscle to contract every 13 milliseconds. And every 13 milliseconds sets it up to where I ask the muscle to contract. At the second it's finished all of its relaxation and it's back to its steady state, it's contracting again. And what will happen is the eye muscle will increase a little bit every time it's asked to contract. Okay? And that is called Trep. And your book has a pretty decent picture of it, right? Now, if I start to make the frequency of stimulation happen while I am in relaxation, I am going to cause 2 things. First, I'm going to cause wave summation. And wave summation is that the force production is going to come up, and it's going to come up higher than it did in Trep, and it's going to, with continuous stimulation, continue to rise to a certain point. Okay? So I will get what is normally asking the muscle to produce a certain amount of force, I will get it to come up to higher amounts. And I'm not changing the muscle number, I'm not changing the muscle sarcomere numbers or myofibril numbers. All I'm doing is in the same muscle fiber, I'm asking it to contract at a frequency that before the muscle technically gets to relax all the way, it's being asked to contract again. And so wave summation is that stepladder approach of force production coming up. Incomplete tetany is when I finally reach that maximal amount of force that is achieved with the frequency rate being whatever it is and the wave no longer continues to rise. Okay? Now, to find the maximal amount of force that I could get a given individual muscle fiber to produce, I need to generate the frequency of asking that muscle to contract so fast that the muscle never actually, in its twitch, goes into relaxation. So I need the twitch rate, the frequency, to be such that the next stimulation comes to that muscle before the muscle starts to relax. And if I get the frequency rate to that point, you never see any hills and valleys because you never actually let the muscle plateau. The next stimulation takes the muscle to another contractile cycle, and I will end up making this contraction come to a height, and I will get that muscle to hold that tension and hold that force production with that high frequency rate. And that's called complete tetany. Okay? So the point of the matter is I can, 1, learn about my muscles in what does an individual muscle fiber generate for force production, how much tension can it produce when I ask it with an electrical stimuli by a button or acetylcholine released. I can make that muscle give me a little more force production with a few little manipulations. One being, change the sarcomere starting position. Maybe optimize it by getting it to be a little bit more stretched out. Or change the position to where it's not as optimal for force production, and so the twitch that I'm going to produce with a less optimal sarcomere position will not be as strong. Right? The next thing I can do, again, in that given muscle fiber in my bath, is ask it to give me multiple contractions. So ask the muscle fiber not to contract just once, but to contract 7, 8, 10 times. But the time of how the next contraction happens is going to be progressively asking it to give me the next contraction with less rest time, with less time in recovery, with less time in relaxation, and in some cases no relaxation at all. And in doing so, I actually do get the muscle to give me more force production, but it's limited to a point. And that point, again, comes back to the sarcomere at some point is no longer going to have an ability to shorten. There's potentially no more actin and myosin to recruit and interact, and there's no more sarcomeres, myofibrils to utilize to make force production. All right, so what do you need to know? The terminology for skeletal muscles, when I ask it to produce one muscle cell, produce a force that I want to measure, is called a twitch. The tension produced by that muscle when I ask it to contract is its twitch. In the twitch, there's periods of calcium being released, the number of actin and myosin interacting. That's the period of contraction. And then there's the period of relaxation when the calcium is starting to be put back in the sarcoplasmic reticulum and less actin and myosin, less room to slide. When I try to get the contraction to be multiple times on that muscle cell, and I ask it to contract a second time the exact millisecond that the muscle finished its first twitch, I am going to induce Trep. And I'll induce the force production for that muscle in its second and in its third contraction to be a little bit more than what it originally was in its first contraction. The increase is very slight, but it is a little higher. If I want to get the muscle to give me more contraction, I want my frequency then to start pushing the muscle to give me a second or a third or a fourth contraction the second it's in relaxation. So it started its twitch, it's got through its contractile peak, but as it's starting to put calcium back in the sarcoplasmic reticulum, go back to its sarcomere resting level, I ask it to contract again. And in doing so, I get wave summation, and eventually I can see a fixed kind of maximal force production called incomplete tetany. If I want to achieve the absolute highest tension that I possibly can from a given muscle cell, I have to contract it in a very fast, high frequency rate, so that way the muscle cell never enjoys a relaxation in its twitching, and in a few twitches, I will reach a maximal force output, but the force produced is never going to show hills and valleys, and it's called complete tetany. Okay? So you need to know those words, know those definitions and what they mean. All right? Now, starting to put, I don't have one muscle cell in a organism, in a muscle organ. So I have multiple cells. And each cell is tied to a neuron. And the neurons might control 10 muscle cells, 20 muscle cells, 100 muscle cells. And the neuron and all of the muscle cells it controls is called a motor unit. And within, let's say, my bicep muscle, I have 1,000 muscle cells. Let's say 100 muscle cells for every motor neuron is a motor unit. And I have 1,000. So I have, let's say, 10 motor units. So if I ever want to ask the bicep to contract, I could ask one motor unit and 100 muscle fibers to contract. And if that's not enough force production, I can ask 2 of them to work together, and now get 200 muscle fibers to contract in unison. Or two motor units, so 200 muscle fibers. If that is not enough force produced, I can ask 3. All right? And let's say I ask 3 and I get 300 of the muscle fibers out of 1,000 contracting, and that's enough force to lift my phone from the table. Okay? That is kind of how individual muscle fibers being told to contract by their motor neuron work together to produce at an organ level a certain amount of force. If I need, instead of my phone, I need to pick up my textbook. Now maybe I need 5 motor units, so 500, half of the muscle fibers in my bicep to be able to lift the book off the table, versus I only needed 3 motor units and 300 muscle fibers when I was lifting my phone. Okay? So that is how it starts to work when we're asking the muscle to produce a certain amount of force, and it needs a certain amount of muscle fibers, and the muscle fibers are controlled by so many motor units, and I'm asking then so many motor units, a sufficient amount, controlling a sufficient amount of muscle fibers to then contract in combination and generate that force. Now let's say I want to pick up my phone off the table and I want to hold it 3 inches off the table. All right, so the phone is now being held in my hand, and my bicep is maintaining a certain amount of force production to keep the phone from falling back down on the table. Now I need maybe those 3 motor units at a given second to contract, but when I ask muscles to contract and they twitch, they go through contraction and relaxation. So how is it that I'm holding the phone, but it's not maybe oscillating up and down as my muscles are contracting, relaxation, contracting, relaxation? This is another thing that the motor units and the coordination of the motor units working together do. So in this picture, over here, consider this first I pick up the phone and I use 3 motor units, and then I hold the phone off the table. Well, when this first grouping of motor units have the muscles go through contraction and then they start to relax, a second kind of tier, so 3 motor units contract, 300 muscle fibers contract, I pick the phone up. As those motor units and those 300 muscle fibers start to go into relaxation, another 300 motor units or muscle fibers from 3 other motor units are going to go into contraction. And when they go into contraction as the other motor unit is going into relaxation, they kind of compensate with each other. So I can hold the phone in my hand off the table, maintain the force production of 300 motor units, but then those second groupings of 300 motor units are now having, or motor muscle fibers are having to go into relaxation. So I get my third group of motor unit and muscle fibers to start contracting. And so what you see is anytime you just ask like to hold the push-up position, your push-up is being held because your muscle, which may be 1,000 muscle fibers, is being put into groups of 3. All right, group A, group B, group C. And in group A, they start the contraction, and as group A starts to go in relaxation, group B starts contracting, and then as group A finishes relaxing and group B is starting to relax, group C starts to contract. And so the minute group C then gets into its starting to relax, group A starts to contract again. And so holding that push-up position is a process in the muscle of A group contracting, relaxing, and B group contracting, relaxing, C group contracting, relaxing, A group contracting, relaxing, and they cycle, right? And so the end result is I'm holding my body in a contraction that's stable, but I'm doing it because the muscle fibers are taking turns, each of them holding the contractile phase with a sufficient amount of force that I'm able to keep myself in a push-up position, hold my phone off the table. Now, at some point, if I start to not have enough force production because my muscles are beginning to fatigue and tire, and I'm not able to maybe recruit as many of the muscles, I'm going to start to shake. And you're going to see the shaking is when, hey, I don't have 3 motor units anymore. All the muscle fibers are now condensed into 2, and having 2 is just not quite enough muscle fibers because so many of them are out of sync and no longer ATP available and messed up and creeping out with their actin and myosin, and I'll start to see the oscillations and the shaking, and then eventually I'll fail, and those muscles won't be able to produce the force to hold me up, and I'll fall down. Okay, and so that's what starts to happen. Again, it starts at a single individual muscle being told to contract, calcium release, actin and myosin. Well, when I need to use more than one muscle fiber, I recruit a motor unit, a neuron, and all the muscle fibers it controls, and maybe I need 10 motor units for a given muscle or 3 motor units, you know, to have enough muscle fibers to pick up the phone versus pick up the book, which is heavier. And then if I want to hold that position, hold that weight up off the table, hold that weight in my hand, hold my position of the body in that contractile state, I can also start to let the muscles of the arm, the muscles of the agonist, set up like a little cycle, set up like a little windmill of each of them take their turn to contract, and as soon as one goes into relaxation, another group contracts, and they can maintain the contraction for as long as they are able to, okay? And so that's what this is showing you in a few ways. One, it's showing you that if I just need enough force that requires just the orange motor unit, so just the 5 muscle fibers that the orange neuron controls. All I'm going to do is ask that orange neuron to release acetylcholine on its muscle fibers, and those 5 muscle fibers are going to contract, and in that contraction, they are going to generate a force, and it might be enough to pick up a piece of paper, okay? If instead of a paper, I'm going to pick up my book bag full of books, it might be that I have to recruit the orange and the purple muscles to work together to do that. And I send all of the orange and purple neuron axons to release acetylcholine in unison. All of those muscle cells contract together, and I now generate enough force to pick up a backpack. Okay? And if I had to hold the backpack in my hand, so imagine you're standing up and you have a backpack in your hand, and you want to hold your hand at a 90-degree angle at the elbow. All right? Now, holding the backpack is pretty heavy. And so let's say to hold that backpack, I have to set up my bicep muscle to be able to maintain a 90% bend in the elbow and hold that force so that there's 3 groups of motor neurons. Not 3 groups of motor neurons, but 3 groupings of muscles that are going to work in unison together. And so all the motor units for group A contract, and then B supplement when A starts to relax, they start to contract, and then C. And I will hold that backpack as long as the oscillations of A, B, and C group contracting generate enough force for me to hold that 90-degree position in my elbow. If it ever starts to not be enough force, my elbow will start to turn into 100, 120, 140 degrees, and then maybe get back to its full extension, and I'll maybe even drop the bag out of my hand. Okay? Now, to do maximal tension, you ask every motor unit to contract in a given second. Okay? And to ask a maximal tension for an organ, it might be that getting every single motor unit to contract in unison might not still be enough force to maybe lift the table off the ground, lift my desk off the ground, okay? And if it is enough force, then I am going to employ every muscle fiber to contract in unison, and I will then generate maybe a contraction and a movement and a shortening of the muscle, okay? Now, when I start talking about sustaining tension, it's easier to sustain tensions longer that are less demanding of muscle fiber numbers. So it's easier for me to hold my phone at a 90-degree angle in the elbow in my hand than it is for me to hold my book bag. The phone maybe allows my bicep muscle, instead of forming group A, B, and C of motor units that are circling into contractile relaxation phase, maybe I can form 6 motor units, 6 groups. And in 6 groups, I can go for hours because there's more recovery time for each group to reset, reestablish cytoplasmic norms, ATP availability, fuel availability. And so holding my phone in my hand at a 90-degree angle in the elbow lets me do that maybe 2-3 hours before the muscle would fail. But holding my extremely heavy backpack in my arm at a 90-degree angle because I only have enough muscle fibers in the bicep to form maybe 3 groupings, it's going to fail in 30 minutes or 20 minutes, right? So for sustained tension, the less the amount of tension you need to develop, so the more groups you can kind of make for sustaining that tension means it's a longer period of time you'll be able to hold that contraction. Okay? All right. Now, most of our muscles have a level of contraction every second of every day. So even though right now you might be sitting on your butt, there is a motor unit and a few muscle fibers in your quadriceps contracting, right? And so that small amount of motor unit muscle cell contracting is what we call muscle tone. All right, and all of our muscles, as long as we don't have a severed spinal cord, all of our muscles at every given second have a small percentage, maybe 5 muscle fibers, maybe 100 muscle fibers, maybe 2 muscle fibers. But there's a small percentage of muscle fibers at any given second that are going through a state of contraction and relaxation. Okay? And it's going to be creating then that the muscle has a certain amount of resting tension, force, kind of oomph to it. All right? If the number of muscle fibers contracting in a given second increases, our muscle tone would increase. All right? So in the quadriceps, let's say normal muscle tone is at any given second 2% of all of the muscle fibers are contracting. But let's say for some reason your quadriceps start having 10% of the muscle in contraction constantly. You would then consider yourself having a high muscle tone in that muscle. All right? On the flip side, if you need 2% to be normal and you get 0.1% of the muscle or 0.3% of the muscle at any given second in a state of contraction, you would have low tone. People who are paralyzed have no tone because they no longer have the spinal cord telling a few motor units, a few neurons to cause contraction in a rotating continuous basis. Okay? Other diseases and conditions, we see changes in muscle tone. So people with cerebral palsy have, in many cases, some areas of their body where the neurons talking to a muscle group are a little overproducing the output and causing more than normal numbers of muscles at any given second to be in a state of contraction and maintaining a state of resting tension. And so they are high tone. And because of that, sometimes they get stuck and they can't relax their biceps to be able to unbend their elbows, or they can't relax their hamstrings to be able to bring the knee out of flexion. All right? And so we see that high tone makes it very hard then for these people to relax. And so Botox is actually used and injected into these folks to try to prevent acetylcholine release, to prevent the number of neurons stimulating muscle fibers to contract. And so then when they're not contracting the muscle numbers like they normally are, the cells can go into more relaxation, less cells are contracted, and the body then can actually bring the muscle into a more relaxed position, a relaxed posture. Okay? And then on the flip side, not having enough tone, you might not be able to, like the baby over here, hold the head up, hold the body position, all right? Hold your posture. And again, people with spinal cord injuries, this is part of the reason why they might end up in a chair, and if the injury is pretty high in the neck, they might have to have their head tied into their chair, held up, because if they didn't, they don't have enough muscle tone and muscle strength and control in their neck to be able to not keep their chin and their head from falling forward or to the side. Okay? So muscle tone is, going back this slide here, is that every muscle at every given second has a few muscle fibers in a state of contraction. Normal amounts is just a little bit. But that little bit allows you to, when you kind of push on your muscles, they have a little oomph to them because a small percentage of the muscle fibers every second of every minute of every day is in a state of contraction. And so it means that the muscle has a few fibers that are kind of pulling on its insertion and its origin spot. It's not enough to maybe cause movement. It's not enough to make the joint stick in a certain position, but it's just enough to where there's some muscle activity going on, okay? And again, muscle tone, normal amounts, the muscles contract, calcium release, actin and myosin binding, sliding, ATP expended, relax. And the next group of muscle fibers starts to contract. And so there's a constant, like, every muscle fiber, maybe in a given hour, every muscle fiber goes through one contraction, one relaxation in your quadriceps. And then the next hour, again, as long as you're not using your quadriceps, every muscle fiber, because its motor unit's going to be in the cycle of, you know, telling it to contract, are going to have a twitch that's going to maintain resting tone in that hour. And everybody does their turn. Okay. Again, muscle tone, too much tone is a bad thing, too little tone is a bad thing. Most of us, we are in the middle. If we have too much tone, there's things we can do. We either learn to live and control ourselves with this greater tone, or now we do Botox and other things where we try to stop acetylcholine interacting with the muscle fibers, stop or mess with calcium availability so we can get relaxation to occur. If we don't have a muscle tone, we either put electrodes on there and try to stimulate it with exogenous electricity, or we try to train the body with physical therapy, or we learn to live with it like in a wheelchair with braces and supports. So not having the muscle ability to make that posture and position is okay because we can hold it up with other supports and straps. Okay. All right, that's a lot of information. Again, it was really in the gritty details of what is excitation contraction, how does that then look as an individual muscle fiber and amount of force it produces, so a twitch, what are the components of when a twitch is made on a graph, how to make a twitch go up in force production without changing frequency by just changing resting position, and then when you change frequency, how to see it go up and what are the different waves, Trep, wave summation, incomplete, and complete Tetany. All right, gang, I know that's a lot. There's a lot in this chapter. Just keep reading, keep working, and we'll get that last, hopefully, muscle video soon.